kingdom Animalia. I hope you are able to recall from class 9 what is cell wall, what is eukaryotes, what is prokaryotes. Further in the clip, I given a difference between eukaryote and the prokaryote so that you can understand or you can remember what's the difference between prokaryote and the eukaryote. Prokaryote which don't contain a proper nucleus, eukaryote contain a proper nucleus. Absence of membrane cell bound organelles, presence of membrane cell bound organelles. So you can open your class 9 book also to recall few of the points. Okay, now we are going to study our 5 kingdom classification in detail. You can see I have mentioned in this clip few terms I mentioned. For with kingdom fungi, myota, with kingdom plantae, metaphyta, and kingdom Animalia, metazoa. These are few of the terms that are asked in your NEET exam. So please go through these terms. These terms are as a synonym of these kingdoms. So these are used asked in your NEET exam. For now we are going to study kingdom monera. In kingdom monera we are going to study 2 bacteria, that is Archaebacteria and the Eubacteria. Archaebacteria mainly contains the methanogens, halophiles, thermophiles, and the Eubacteria mainly contains our various types of you can say Cyanobacteria, nostoc, and amina. You are going to study each and everything in detail, and these are quite asked in your NEET exam. Now again a diagram is given over here, and please keep a thing in your mind that in kingdom monera many bacteria are included. So in kingdom monera it divided into Eubacteria and Archaebacteria. Eubacteria are also known as true bacteria. Further it is divided into heterotropic and autotropic bacteria. In autotropic bacteria it is divided into chemoautotrophs and the photoautotrophs. Chemo means which use chemicals to synthesize their food, and in photoautotrophs means which use sunlight to synthesize their food. Okay, in Archaebacteria we are going to study 3 main bacteria, that is halophiles that are found in the salty areas, thermoacidophiles mainly which are found in the hot acid area, and methanogens mainly which are found in the marshy area or inside the ruminant gut of animals. What is ruminant gut? Means inside the small intestine of cows and buffaloes. Okay. Now look at the next clip. Before going in detail, we are going to study simple characteristics of bacteria. First of all, they are known as prokaryotes. They are unicellular. No membrane bound organelles are present in these bacteria. They are single cellular chromosomes. They have single cellular chromosomes, and reproduction is mainly done by binary fission. It means a single parent is divided into 2 main progeny, that are the daughter cells. Okay. Now look at a typical bacterial cells. It contains pili, or you can say your fimbriae for your flagella for your movement. The cell wall mainly provides the structure, the support. Cytoplasm, fluid part in which the cell organelles revolve. Ribosomes make protein, and the chromosome is the cellular DNA for the genetic information. Few more characters are also mentioned, that they are present numerously and widespread organisms on earth. They include the smallest and the simplest life form, mainly unicellular, and they mainly live in clusters. Means they like to live in groups, they don't live singularly. Moreover, only kingdom made of prokaryotic organisms, the only kingdom made up of prokaryotic organism, and they don't have a well-defined nucleus. They have a nucleus but not a well-defined nucleus. Okay, now we are going to study few of the shapes that bacteria contains. There are various shapes that are present of bacteria, but mainly we study 4 to 5 shapes that are mentioned in the syllabus. That is a cocci, bacillus, spiral, and the vibrio shape. Okay, and these are the most abundant bacteria. They can be present anywhere. They can be present on your phone, they can be present on your head, they can be present on your skin, they can be present on your wires, they can be present on your fridge. Everywhere the bacteria are present, and they are divided first of all mainly on the basis of their shape. Further, they are divided into autotrophs and the heterotrophs. Okay, now look at the shape that you are going to see through a small diagram so that you can easily understand. Cocci, or the spherical shape, bacilli, or your rod shape, spherical, okay, that are known as spirula, having flagella for the movement, and the coma, or the vibrio. Next, you can easily understand the various. I have provided 2 to 3 clips so that you can understand the shapes of the bacteria quite properly. These are the shapes that you are going to study, and on the basis of shape also we have divided the bacteria, but it's not mentioned in your syllabus right now. Now, kingdom monera is basically divided into you can say two groups, that is the archaebacteria and the eubacteria. First we are going to study in detail the archaebacteria. Archaebacteria are the primitive forms of bacteria that are present on the earth, means they are the oldest bacteria that are present, and they can survive in the extreme condition. Extreme condition, they can survive in extreme temperature, that's why they are known as thermophils. Extreme salty areas, that's why they are known as halophiles. Acidophiles in various acidic condition, various basic condition, and in mainly they are also known as methanogens, matlab they contain the methane gas also, like your gobar that is used for the production of methane. I would like to recommend you to pause each and every clip so that you can understand what I'm saying to you. Again, this clip symbolizes that there are various bacteria that live in salty areas, hot springs, marshy areas, and archaebacteria differ from other bacteria and having their different cell structure, and they are responsible for survival in the extreme condition. Methanogens are the present in the gut of several ruminant animals like cows and the buffalo, and they are responsible for the production of methane or the biogas from the dung, means gobar of these animals. Okay, now you look small clips, pause the clips, and you can see properly what I have spoke to you. Divide into two, archaea and the eubacteria. Archaea are the ancient bacteria, okay, and the eubacteria are the true bacteria. Further, in four categories it has been divided that we have studied. Methanogens found in the gut of the, you can say, in the ruminant animals like cows and buffaloes. Thermoacidophiles mainly found in hot and acidic waters. Best example is of this Yellowstone National Park in America. Next is your chemosynthesizers. They are mainly found in the hot sulfur vents in the ocean floor. Okay, next is your extreme halophiles, mainly found near the sea water that have the salt. Best example is your Dead Sea. Then the relationship with the humans is quite important point, as we can see, we have observed that there are few bacteria that are useful to us and few bacteria that are harmful to us. Useful bacteria, must one major bacteria, that is your lactobacillus, that is used in the production of curd, and with the help of this bacteria we are able to digest our food properly. But there are few bacteria because of which various diseases are caused, like your disease like cholera, typhoid, they are caused by various organisms, various bacteria mainly. And bacteria, if you remember, your typhoid, salmonella typhi, your cholera, your vibrio cholerae, these are the mainly, you can say, bacteria that cause the various diseases. So you can say that the bacteria be both useful and harmful to humans. Okay, now the next topic we are going to start is our eubacteria. That are true bacteria, mainly contains of two parts, true bacteria and the cyanobacteria. Now we are going to study each and every point in detail. Eubacteria, largest group of monorons. Majority of bacteria it contains. Most of them are decomposers, heterotrophs, and saprophytes. Some are photosynthetic older, like your cyanobacteria that we are going to study separately, or your chemosynthesize that use various chemical for the synthesis of the food. Both these we are going to study separately. First of all, we will study true bacteria, then we will study cyanobacteria. Now, first of all, eubacteria, the true bacteria found in numerous environments such as the surface of human skin, intestine of animal, or on a wood log, and they are often identified by gram staining method, gram negative and the gram positive. This we are going to study further in your chapter number 9. Okay, now the few more characteristics we are also going to study, that they absorb food molecules from their surrounding, they rely on other organisms their food. Mainly they are divided into two, decomposers and the saprophytes. They can be beneficial for the humans. For example, in the intestine, mainly your E. coli bacteria helps in the digestion of the food properly. Okay, now next we are going to study our cyanobacteria. Cyanobacteria mainly lives in fresh water. It contains chlorophyll and produce their own food, different shapes and attached to each other in long chaining clusters. Example is of cyanobacteria are the nostoc and the anabina, and they are the green scum, you can say kai, that are found on the top of the pond or the lake, and you can easily observe by going over there. Here are the few clips. So it's in a microscopic clip of a nostoc, and they also help in fixing of the nitrogen, as they contains a structure known as heterocyst. Heterocyst helps in the fixation of the nitrogen, and you can see few pictures what I was speaking about. Mainly it is called kai, but it's not kai, but it's mainly the cyanobacteria that is present on the surface of the pond or the lake. In the end, I would like to conclude with a summary of the eubacteria. Go through this summary as it will be able to recall what I have taught to you. mainly eubacteria and the cyanobacteria are quite properly mentioned. Pause that and study it properly. Last topic that is mentioned is your mycoplasma. Mycoplasma are the smallest living cells that are mainly discovered. Mainly PPLO is the best example of it. What is PPLO? PPLO is a pleuropneumonia like organism that can survive into without mainly oxygen, and they are mainly pathogenic, causing disease. And I hope this is clear to you. Tomorrow again we will meet with the next topic.